Welcome to the Learn Biology podcast, where we break down complex biological concepts and make them easy to understand. Today, we'll be beginning our series on cells. In this episode, we will be discussing cell theory, restrictions on cell sizes, types of microscopes, and a comparison of prokaryotic and eukaryotic cells. Let's start with cell theory. Cell theory states that all living organisms are made of cells. Cells are the basic unit of life, and new cells are formed from existing cells. This theory, developed in the mid-19th century by German biologists Matthias Schleiden and Theodor Schwann, has become the cornerstone of modern biology. Next, we'll discuss the size of cells. Physical limitations impose a maximum size that cells can achieve. As a cell grows larger, the ratio of its surface area to volume decreases, leading to difficulties in exchanging materials between the cell and its environment, such as food, water, and nutrients. To maintain effective exchange, cells are limited to smaller sizes with a higher surface area to volume ratio. Scientists often use microscopes to observe cells in greater detail. Two major types of microscopes are light microscopes and electron microscopes. Light microscopes use visible light to create images of specimens, while electron microscopes use a beam of electrons. Light microscopes have a lower resolution and are used to study living or stained cells and tissues. The resolution of a light microscope refers to the level of detail that it can capture in an image, and can allow for distinguishing two separate parts of a specimen or sample. Electron microscopes, which come in scanning and transmission forms, have a higher resolution than light microscopes and are used to study surface and internal structures, respectively. Finally, we'll compare the two main types of cells: prokaryotic and eukaryotic. Eukaryotic cells, found in plants, animals, fungi, and protists, are more complex and have a nucleus surrounded by a nuclear envelope. Prokaryotic cells, on the other hand, are simpler and lack a nucleus, but have a plasma membrane, cytoplasm, nucleoid, and ribosomes. Some prokaryotes also have a cell wall, flagella, or capsule, which are characteristics that help distinguish these microorganisms. In conclusion, this episode provided an overview of the basics of cells and cell theory. We discussed the restrictions on cell sizes, the types of microscopes used to study cells, and the differences between prokaryotic and eukaryotic cells. In future episodes, we will delve deeper into the intricacies of cells and continue to build upon this foundation of knowledge. That's all for today's episode of the Learn Biology podcast. Don't forget to subscribe and leave a review. See you next time.